Hello everyone. We had already discussed a conceptual understanding on Lac Operon in some of our previous video. So this particular video is a continuation of that, where the basic conceptual questions that arises on Lac Operon based on NCERT will be discussed. So for you to understand this particular questions that is going to be there in the video, so you are requested to have an idea of what Lac Operon is. So those who have not watched the previous video, so kindly watch and join us in this video, right? So the first question here is, what is the ultimate use of Lac Operon? So you know that Lac Operon is mainly meant for regulation of three genes: Z gene, Y gene, A gene. Okay, these three genes are going to code for the respective enzymes that is required for the metabolism of lactose. Okay, metabolism of lactose. Now the question is, what is lactose? Okay, lactose is a disaccharide. Okay, meaning a sugar which is present in milk. It is a milk sugar. Okay, disaccharide meaning it consists of two sugar. One is glucose, the other one is galactose. So it is a combination of glucose and galactose. So ultimately the purpose of Lac Operon is these three genes, ZYA gene, should be expressed only when there is a lactose present. Okay, to metabolize the lactose into glucose, to break down lactose into glucose and galactose. And if lactose is not there, this three genes should be shut off. Why? Because the expression of these three genes requires energy in the form of ATP. And if these three genes are getting expressed in absence of lactose, where the enzyme does not have any role there in absence of lactose, the cell is going to use its energy without any purpose. So ultimately the idea is, when there is a lactose, these three genes should be kept on, or it should be kept off, right? So that is the ultimate purpose of Lac Operon. So this answers your first question. Okay, right. Second question here is, what is the use of trans-acetylase? Right, for this you have to know that, as I have explained, three genes: Z gene, Y gene, A gene, or alpha gene. So what does the Z gene codes for? Z gene codes for beta galactosidase, right? It is what is required to break down glucose and galactose, right, which is the main enzyme. And what is this Y gene? It is a permease. That is, whatever the lactose that has to enter from outside into bacteria cell, okay, will be helped by this particular enzyme, which is permease. Okay, permease, which is coded by the Y gene. And what is this A gene altogether? It is trans-acetylase, whose function is not there in the NCERT. So these two function, okay, the function of beta galactosidase coded by Z gene, permease coded by Y gene is there in the NCERT. But what is the purpose of this trans-acetylase, which is coded by the A gene, is not mentioned. So A gene is trans-acetylase, which prevents basically a beta galactosidase from acting on other sugar other than lactose. Why? Because sometimes the product formed when beta galactosidase act on other sugar is going to be toxic to bacterial cell. So that is exactly prevented by the trans-acetylase, which is coded by the A gene, right? I hope this question is also clear. Okay, right. And next question is, why is a glucose preferred carbon source? This is there in the NCERT. Glucose cells, E. coli cells or bacterial cells prefers glucose. If there is no glucose, then lactose is preferred. Why is that so? Because glucose is already a simpler form of sugar. Okay, so it can directly enter into glycolysis. Okay, rather than a lactose, which has to be broken down to glucose and galactose, which requires extra energy in the form of ATP. Glucose being the simplest form of sugar, okay, no requirement of this beta galactosidase, permease, trans-acetylase, those enzymes are not needed. Okay, so directly the glucose can enter into the process where ATP will be generated. So it is the simplest form of sugar. No extra metabolic pathways are needed for its formation. Okay. So that's the reason why a cell or a bacterial cell here prefers a glucose as a glucose initially. Okay. Right. And this question is, why is a Lac Operon referred as a negative regulation? It is referred as a negative regulation because the repressor molecule, which is coded by the I gene, is going to prevent the transcription of the 3 genes. Hence it is called as a negative regulation. Okay. So I repeat again, the repressor molecule coded by I gene is going to bind with the operator, thereby it is going to prevent the transcription of ZYA gene. Hence it is called as a negative regulation. I think this is also very clear. Okay. And why is lactose referred as an inducer? Obviously because once the repressor binds with the operator, okay, polymerase cannot reach the structural gene, that is ZYA gene. So the transcription of 3 genes are prevented. Now what is making these 3 genes to get expressed? It is the lactose. Once it enters into the cell, okay, it forms a complex with the repressor. That complex cannot bind with the operator. So lactose induces the expression of ZYA gene. Okay, hence lactose is commonly called as an inducer. See here also one more thing. The permease is what allows the lactose to enter from outside to bacterial cell. So the permease, there is always a basal level of expression of these 3 genes, and that permease is needed initially for the lactose to enter from outside to inside. Then only the lactose is going to act as an inducer. Then excessive expression of these 3 genes will happen. Okay. Initially the idea is a minimal permease, okay, is required for the transport of lactose from outside to inside, for which even without lactose there should be a basal level of expression of these 3 genes. Okay. I hope all these questions are well answered. If you have any other query, you can drop us, okay, in our mail or in comment section. We will try to reach you with a solution. Okay. Thanks for watching.